welcome back.
Yes.
You'll have a good break? Excellent. Me too. I refresh myself. It's very hot. So again, if we lose power and it gets really hot, we can always go out, get some water, and then come back. I want us all to be hydrated, okay?
Person 2 Johnsons, Chilo Shudus, young kid, San Langaso, lost chicken was who chasnaya, Chilo do Chitwando, the China race, Nasukatim, and Azupuna Chiti Guru, Chitonrogis.
Good. So there was a question about DNA and RNA and replication.
DNA, RNA, Negasuge, Dip Karsa, the Suchech, or Tikolaya, George Dus.
The point that we were trying to convey is that replication happens in DNA.
Replication
Replication happens in DNA.
DNA.
But in order for DNA to replicate itself, it requires a primer, a small segment of RNA.
Taku Dreshiti Chialaya, Karagu Dreshimasadakasa, Dreshiti Chialaya, and a cool primer site Guyores. The primer site DNA Chashichiris.
What DNA cannot do is self-replicate without that small segment of RNA.
Tati Kura Kitty Tandaga RNA Chashi Tumbuti Meba Imbana, DNA Kura the Dreshuchia de Unguyo Maris.
So one idea, one theory of how life may have originated is that perhaps it may have begun with a molecule based on RNA with the ability to replicate.
Can you repeat that again?
Yeah, so one of the ideas of early life and how life may have evolved is that it may have started with a molecule based on RNA.
Tampo de Canada uchi chonguyore se tikolaya ane kusugi chigda tatanchi kariyore se stambala tatsuchi kariyore se malaya ninga tsuga RNA la shisha bigin didichini chongre se chkorwa otigi tatsuti yores.
So perhaps life may have begun with RNA and may have evolved into something more stable like DNA.
Chichena ane sesho de tambo de chi D kasa RNA ni gusu ane kuta ko yerji sukor ta penju chungala tene yerji chola tene de ane ko this tembuyo pasukor ta tembuyo pe DNA gi ngula chro tene ins pare chichinas.
So for example, some viruses have either DNA or RNA, and some viruses have double-stranded RNA, which is really weird. And perhaps how life may have started.
Ta Nielsen virus, Nielsen Kachila Imbana, Kashilaya RNA yore, Kashilaya DNA yore, Kashilaya Yanko Nyagma Nidenjerwa, Nyagma Nidenge, RNA Yubayan Tina yore, since Tambalaya Diko, Netan de Dus Kensauchi yores. That's Chichi Bainaya, Dinado, Tangasu, Netan Dinado, Ghanaian Chisho, the RNA Munisagorwa, Sesho de Tambuchon at Dinado in Sabaris.
Good? Chris, anything? We're good? Excellent. All right. So let's continue with our topic. So I was told that I have to point in this direction, otherwise it's not working. And I was going crazy until Tabo told me, and he made fun of me, so I'm sad.
I'm sorry.
All right, so let's take something completely different. We understand transcription, we understand translation. We learned about histones 2 days ago, right? So here we have a set of organisms with a sequence with each organism.
Tatanga Suge, young Tanguela, Desha Juti, this Mandoachi, those Desha Jagis, Tatanda Dinananga, so Histon Chiora, Histon Tiziti, Yamakashing Olangasungo, Tisichi, Yoris, Tatanga Suge, Semche Mando, Kachilaya, Nalaya, and the Histon Digi, Koga, Taquarum Chigora, Pangarum Digandochi, but that against.
You all remember histones, their function is to condense and decondense DNA.
다 단다 체조게 지치나 탱게 그레스 로 다다 가수 히스톤 지지 히스톤 그 젤리티카레스나 가수게 DNA 티 쳇담보주 다 야나 쳇르베지에티 그런 게 젤리티레스.
So what is unique is that if we look at these sequences, is this DNA or RNA? What do you think this is?
다 단다도 스타바인 바이나 그런 게 또아 고파치도 그름치도와 the current data by the DNA inside RNA inside.
Somebody said it. Protein is not DNA, it's not RNA. This is a translated sequence of protein. These are polypeptides.
Right? Tanda chigdela ya didela sonsra de karsa tizirela sonsra de dila maris karsa DNA la mare RNA la mare ko da phabju chi sabigi tisigi thangwa sigro da tizirwa tizidres.
So we have the sequences across these 5 different mammals: humans, mice, rats, cows, and chimps. And you can sort of appreciate that they look pretty much like each other. They are very similar to each other.
Hada dada de la ya nga chuge socha nga to mi tan chewa chi chi pashu chi chimpanzee ku sunga idu sta ba na kogi history no yo pi ge tjigi khorum chi gorwa du sta ba na subject ma no ngwa himana khorum ta ba na halam da son da yores.
And if we compare the actual sequences between each other, we can actually see where there are subtle differences. So wherever you see an asterisk, they're completely similar to each other. Whenever you see dots, there may be subtle differences.
Hada dada de la ya du sta ba na dada de ku khorum de du se dusha yes ro de na ne ani dada de la ya ta nga su karto yo sa sto karma ge ta yo sa ti kala na ko chu cha chong che ani de la ya cha yo sa sto de cha chong mai ba ge ni dres.
So for example, here we have S, A, S. A few differences between the organisms.
Hada dada de tamputa ta ba ne ni S A S re do ko ge samche ma do na ya ku chapatis do.
Same here, we have T, S, S, T, and here we have A, T, A, so one difference, and this one has V, A, so you could see where there are subtle differences.
Hada dada de cha yo sa te la ya ane ko chapatis this yo pa de danda de na ne khorum na ne thong do as.
So here's a question for all of you, and I welcome you to discuss it for about a minute or two. Most of the sequences across the organisms are identical to each other with a few differences. Why is the conservation of the sequence important?
Hada danda de la ya chu rukana ne kamani sam gna la ya tu de chero che swa ne tu de che de danda nga su de ge gang ge history ge de ge khorum de la ta ba na ku khorum chi ge chashi ma chi shi ching song le shas capitis this chi ma shi men do sta draw de ka rin se na ne nga su ge khorum ti nyer so che chu ro conserve che chu ro nyer so che ti kari chi ne ke chen bu res.
So discuss it for about a minute, maybe 2 minutes, and then we'll maybe hear from a group or two on why you think this is important.
Hada rukana la ya kama chi ni chi kren la ya tu de chero ches ne ti je la ya ne rukha chi ni chi la ya ngutu che ku ka nangis.
Okay. Does anyone want to share with us why you think conservation of sequences is such an important part for these organisms?
다 되고라 무엇인지 사다 징외 징외도 줄라 따바인바나 소소도 줄라 따바인바나 가수게 그름띠냐 소시야지거와 데 가르치는 게 전부래 진짜 시작지다스
단다된 코란데 냐짜제 기침보는 배치는 대인사를 정도 가르르스나 단다디게 냐짜 따다 야바 마자네 니츠데 따다 좀 마토 디옹게 아니 응저 단다 캔서 차다도 정말 이제 용거다 저 디니츠되게 강하되게 되게서라스
I think the importance of the conservation of the sequence is to relay the information consistently from one to another. Otherwise, we have like a cancer or other diseases like that.
Round of applause. Excellent answer. Right?
Very
good. Very good. What do histones do? Histones allow us to express genes. If we change too many things on the sequence, the histones become non-functional.
다단다데 램페야 보나서 다 히스턴기 그런 게 젤리티 가르치기서나 가츠가 르지기 띵 원남티마 토엘라야 로파치기요레 로파치기요레 스틴스 담발라야 케티 아니 가츠기 히스턴기 나나요 피고 코름티라야 지라 마푸치 땅바이마네 케티 지라 마푸치 테바이마나 아니 히스턴디기 젤리티 타타 치트카마레스.
And the question is, if we start to accumulate a lot of mutations and we change the sequences of histones a lot, they frequently become cancerous. The gene becomes non-functional and we start accruing cancer within the organism.
다다다데로아 케티 아니가 속에 히스토나 요피게 코름데라야 타코 안지라시키 첨부치 테바인바이나 아니 그 젤리 티타타치 마트와트네 가서 르지 원음 티타탄고 마트바데 쮸엔치데데 아니 가술라야 아니 디네디 종구요레스.
And a lot of research has gone into understanding where in the sequence of histones do we have variation and what type of cancer is associated with specific portions of the sequence.
다다다 가서 히스토기 그런 게 그런 거나네 간기 지라야 아니 지가서 다 만도 쉔셔바다 딜라티 차시 간기라야 쉔셔와 딜라테네데데 아니 가서 디네게 리 간기 종구요 메지거와 티콜라야 아니 심지디 마부지요레스.
So for example, you may have your sequence of histones and if you have mutations in this particular portion, in this location, this is associated with bone tumor or brain tumor or specific kinds of tumors within humans.
다다다 피체샤바이나 가서 히스토그나네 코랑구나네 칸치라와 디치라야 디 닌저 총바인바이나 닌저 총바딜라테네데테 ku kangi tandawoyoresena asugi rikoki dine chongata yana de kasa lepagi tene chongachigoro titandawoyores.
So it is very important that histones playing such an important part for cells and cell expression that they be conserved and they remain functional.
That is Tamalaya Tanda Histon to the Tigi and Asuga Tapunganane Riziti Kandas Mangote Laya and a Kogi Chilite Druki Sanwa and a Korumti Taku Tembugan and Yersociate Hajikachaburis.
And a lot of research has gone into looking at this specific type of pattern, looking at mutations in histones and the type of cancers that are associated with those mutations.
다다다 데캅스와 히스톤기 코랑관아니 간기 가와코알라야 니 가와코알라야 닌지종샤 닌지종아 텔라테네 데타 제네기리 카리카리 종샤세 지티쿠 파롤라야 데와티 콜라야 데와투피 콜라야 아니 tak시 심지티 mampo치요레스.
Good? No questions? You have a question? Okay, yeah.
So is histone a gene, a part of a gene? Because, you mentioned that when there is abnormal expression, then there are different cancers. So is histone
Well, so there
a
are
gene?
genes that express the functionality of histones, right? Histones function properly based on the normal sequence within a specific location of the sequence of the protein, right? So there are certain proteins that will express the normal function of histones. If we change that, if we mutate that, then the histones become abnormal. Does that make sense?
His question is histone a part of a gene? But it
histones
affects the
part
expression,
of
right?
a gene. Histones are not genes. Histones are a set of, how would you say this? Try to think of a good way to say this. Histones are products, right, that allow the DNA to be
Expressed, right?
to be crumbled and fit within the nucleus, right? The presence of a histone, the emergence of a histone depends on the normal function of other genes, right? So some genes are going to express the normal function of a histone, but histones are not genes. Does that make sense? Yeah.
Hata dada gela ki draw the system balaya tanda degi ngasu histone la ya ko nyinji tabai bana sro ata ko chunchi shor bai bana ngasu ke tene chongo yores gore sta ti bai bana ngasu ke histone se te rizgi ki chashi roes histone se te rizgi ki chashi mares histone se te ngasu ke rizgi te ko chatam de zoa ta cha lot zoa ge ko cheli chen te ro imbai na yans histone khorang ge chi chechu ngoya te karila ra le gre sena ta rizgi la ra le yorwa ya mare rizgi ngonsula tan de ta ke te histone la chon shor bai bana tila tan de ta ne ngasu ke ta chi tendela sopan jo tende jo a chi gorwa ni tam te jongu yore dikapse histone te rizgi chashi chi maris.
That is a very good question. And
So,
we will
yeah,
go
but
back to what Chris has taught us yesterday with organelles and the distinction between prokaryotes and eukaryotes is that prokaryotes do not have histones. Only eukaryotes have histones.
Da kesa ge Chris la ge ngatsula ya jingwe da jingwe ge thapung na ne thapung ko wang tha zu ma bo de shi ja yores te na ne ngasu ge ningden da ningte ge thapung la ta bai bai na ane ningden ge thapung chi pu la ya ne ngasula ya histone de yores te ma to pi ningte thapung la ya ane histone de yo maris.
Any other questions?
Trajemba yos Ayosa.
What was
Yes, I was asking them to answer, right?
Oh, yes.
Do you have any questions? So they need to answer, like they just.
that? We all need to go to lunch. I know. We're hungry.
Yeah.
All right, so let's spend a little bit of time going through the basics of mutations. Now, Chris tomorrow is going to spend a lot more time, a lot more in-depth into mutations, so I'm going to briefly introduce that to you.
Sanya bana ge Chris lage ninjur kola ya ta jiun de jagres ta tene bana ngasuge ninjur kola ya chu chi de jagos.
Okay? So, let's say we have DNA, right? The complement of DNA, transcription, which is RNA, right? And translation, which is your peptide chain. So this essentially shows the central dogma.
Ta de ngasuge korum de la ta bana ngasuge tawa nyimpu de galay de jayes ta tampo de bana ngasuge da dikinya mater doa tigi opa de dir do pc gi nyang mater doa te tampo de nya ngasuge DNA gi nyang ma tampo tan ba cherwa DNA nyang ma tampo ni bala tene de ngasuge RNA ti ngorwa phap su chi wala tene de RNA gi nyang ma te ngorwa RNA gi nyang ma te la tene de o te kare system bala ne ngasuge phap ju chi de ti kare ngore ngasude le tiji gi thawa te ngorwa.
And you can see each triplet is translated into a different, unique amino acid.
Na hanta danda chesu de la ta bai bai na ngasuge rigzi gi changdar wa ta rigzi gi changda simcheng gi rigzi gi changda re te ane amino chuje te ngi la ya dus phaju chi ba su de la thong do was.
Okay? So I'm going to use the symbols of happy faces and sad faces for mutations.
Hanta danda bai na ngi kare pechu tang gi sena ane ko chi rimush to ga te chi to no yo pa ta true gawa tang sure ge dong pa ge sidwa di pe chu tang de tang ga chu ge ninjur se de som chigins.
Okay, so we have the non-mutated segment. This is perfectly functional. DNA, RNA, polypeptide, non-mutated.
Da de bana ga chu ge DNA re RNA re ane ti ona tzi ban de kap se ko ninjur du yo mares.
So we're very happy, but now let's look at some mutations.
Da de kap se ko ninjur du me sa ko parti ga ter muti ga tele ko ga de no ten de yus ta da de la ga chu ninjur ju ji che ta ge yes.
So let's say we have what we call a silent mutation.
Da ko da de la ya ga chu ge ninjur ga na ne ri ta bu de ne silence se do ga silence se de ta ga chu khusum bu ta ya na shinjam ge ninjur res.
So we have in the non-mutated CGC, in the mutated we have CGG. So that G at the very end of the triplet changed.
Da de na ne ko ninjur ru da yo res ta ninjur ta de shinjam ge ninjur se te da silent ra de ka sim bana ga te la ya ga su DNA na ge te ka re se na CGC ri do wa ta ma yon sta bala ga su la CGG cha do wa de sta bal te na ne sim chen chi ki chen ba tha ma te la ya an jro the de yo ninjur chin yo res.
But because of the redundancy of the amino acid coding, both CGC and CGG, they both codify arginine.
Da de la ya ga so ge ko ka so ta rizgi gi ti khu man khre amino acid la ya phajur chin sta bala ya amino churji kha chi la ya ko rang ge da tin da ge sum sen tin da ma bo do wa ko ma bo do wa ma bo ye cha ko gi thamati jwa chim ba ba na ya ko ni sha ngun ne te ti ka se na ti tzi ti ka chi tzi ta ran ja chi pat ko res.
So we call it a silent because even though we are changing the sequence, there is no impact on the amino acid. It is the same.
Da de la ya shinjam ta ya na khusum se shinjam se ge jun sen ti ka re se na nga so ge rizgi gi ko rang ge kheram te ge thama de la ya jwa du go res. Jwa du go yor da imba na khu ni se ge tzi te ons da bale tzi la ya chapre chong yo mare ranja res tin sta bale de ring de la nga so ge shinjam ge ni jur se gres.
So they're not easy to detect, but they are considered a mutation. So yeah, we're still pretty happy, although we do have a mutation.
Da de kap se ta ko ni jur chi yo res. Imba na khu la ya shu chen chen ba me do wa. De kap se ta dong ba ko chu la member ge ne ta res.
So let's look at other mutations. A missense mutation.
Ta ninjur ge shenda chi ka re se na missense to ga missense la na tun tilwa.
In this case, we compare our non-mutated CGC with TGC. So this T replaced this C.
Ta de ta ba na gang ge CGC ge C chi to ga tang bo te T ge ne ko ji sab chi sha re sab chi sha yo res.
In this case, we call it a missense because now with the non-mutated we have arginine, with the mutated we now have cysteine. So we changed the amino acid. The polypeptide has changed compared to the non-mutated.
Ti ge kap se ko ton thil wa sa go ro ta missense ton thil ba ti la tene de te ko ge ta tampu nga chu ge ninjur ma chong be ge ko ge khoram te la ta ba ba na de la ya chicken jwa do do re chicken jwa do te la tene de te ane ko ge tzi chi go wa tzi te la ya jwa do do wa ris ta ba la ya de kap se nga chu de la ya ton thil be ge ninjur se go res.
So in this case, we're getting into some issues. We may get into some trouble, right? Because we are changing the sequence of our polypeptide. We may change how well the gene or the protein is functioning.
Ta de kap se ba na ga chu la ko ninjur chim ba la ta de ta ko khorum la ninjur chim ba la khojule khorum la jwa chim ba la ta de ta ga chu la ka nge chi thig res ta ko ge kange kadeste gre se na tzi tin jwa do sarwa re mare khore chi jwa la tin de ta tzi la jwa do sa tzi la jwa do sta bala ya ane tanda tzi ge na ne chashi chi la jwa do ra da this sta bala ya ga su ge tzi ge thawa de ge peptide tirwa tzi de ge 아니 젤리라 아니 주라 지치나 드스바레스 디스탄발라야 디캅스 코타 강게 차기오레스.
The third type of mutation, and by the way, these three are known as single base mutations where we change one base.
아하다 단다 디게 리스미스도가 리스미다 리 단다 데시자송아라 인주 느줄라 카르세게르사나 나츠게 지지 칸치기라야 주라침비게 인주세고 요레스다 디캅스 나츠 지지 칸다 디고 코랑고난데 지지 칸치마시 조탄고문도가 레마레 오 디스그레스.
So for the nonsense mutation, we are introducing a stop earlier than it should be.
다들라야 nonsense라는 도메 양나 동로와 도메기 인줄께서 가르치그레스 스탄발라야 나수게 가르치그레스나 다참 조가기 다치거와 참 조가기가 다티 지금 해 말하는 거는 뭐날 해기요레스.
So if we compare the non-mutated TCG with TA, the A changed, G, in RNA, this is UAG. We changed the T to the U, and UAG, as you may remember, is one of the stops.
아하. 다다나 디캅스 바나로와 가출라야 디캅스카레 강강텔라 TCG르도와 아니 오텔라야 TAG로와다 TAG 차고도 단단히 RNA 타바야나 UAG 차그레스 UAG 차바인 바야나 디 가리 차그레스나 타마텔라야 코게 다 상다티 카레스나 가서 참샤지거와 어디게 다드레스.
And the consequence is that this polypeptide is going to be shorter because of the earlier stop.
아하다 코게 참지가게 다테 응원랄 해게요자 근데 단다 데라요 그 뜻이 통화지거와 망도 뜻이 통화지요 바데 젠다 통들바이바나 통화요레스 그참 땡걸라 잡아와.
So again, we are in trouble. It may be likely because we have a shorter polypeptide compared to the non-mutated. This one may lose some of the function.
아하다 단다 가수게 닌줄 마침베기 야마테 따다 닌줄 마침베기 뜻이 땅 dur바임바나 단다 가출라 라비게 뜻이 통화텔라야 통화요로와레 코니지 테데오라 데스탄발라야 옹옹게 땅 dur바임바나 코기 젤리라야 치치나 쇼쇼요 그레스.
So just remember, silent, missense, and nonsense, all three are single base mutations where we essentially replace one base for another.
아하다 단다 데게 지 신점기 닌줄에 아니 돈 투비 닌줄에 돈 도메기 닌줄사 저라 닌줄 섬데 가림방기 구글에서나 지지 치기나네 코르메 까치글은 지로와 땅 발라텐데 데타와 코르 까치기 지우지 발라텐데 데타 종게 바텐게 구글에스.
Let's look at the last type of mutation. There are many. I'm only going to cover these ones, okay? The last type that I'm going to cover is a frame shift mutation.
나하다 타마데 인중 나네 타마데 단다 고토 제티칼라 바이나 friendship 딩가 속에 니메게 돔지스 다 frame system 발라 돔로와 돔 틴주라 땅데 인주 중 아드레스.
Unlike single base mutations, frame shifts do not replace, but rather they are inserted and they shift the whole sequence one spot.
Can you repeat that again?
Yeah, so basically instead of just replacing it, so we have, for example, CGC, we have CTG, but this was not replaced. It inserted itself, pushing everything one spot forward.
다 데데레서 다다 가수가 그 님에 롬지티카레 시스템블 당기 바나 코랑 그 코름 지크차 지글라인 지티 차브지 자고도와 래 말에 지글라인 지티 차브 자고로 이치래 말에 다 단다 데라야 님에 롬지티 바나 지글라인 지 차브 자고요 말에 지금 바로라 어떻게 제 축샤요레스 바로들 어떻게 제 축샤스tambalaya 쿠게 지티 선바 만 버적 당가다 그러고 와 디스tambalaya 쿠 코름 티트쿠요레스.
And if we insert one base, as you could see, the rest of it does not match with the non-mutated.
다 코나 텔레 제지치 빨라 어떻게 저기 스tambalaya 되게 잡아 요피 다 코탄 코가 잘라 요피기 아니 단다게 티치거와 에미노 시리즈치거와 티 닌줄 마총 베기 에미노 시리즈타 베르카가 차거레스.
Essentially pushing everything one spot forward. You can also have a deletion where you delete a base and everything retracts one space back.
that is Tabalaya, and a Tanda de Parola Chikudu, Rabare, Parola suggests Chikuara, young Paroni Tandage Casa de Yandu Tripsashi, Toyatina, Yoris, Super, Maple Shaba in Bana, Tanda de Banaco Chupa and Bana, Mama Nurgudua. Ketichi Toba in Bana, Yakum Dugures, Jalo de Somaya and Gorwa.
But now, if nothing matches compared to the non-mutated sequence, then all the amino acids are going to be very different.
Tanda de Kuranga Santa data by Bana, Kuga Ninja Machim Big Yamate Durban Bana, Kuga, it's how my Mandawar in the Wa. The Mandawa Latin thing, also all a rapid than the amino acids Mandawar.
So the structure, the primary structure of the polypeptide chain is very different. The last few amino acids are very different from the non-mutated one.
So Tama de la Drubig Titsig Tawachigorwa, Titsig Tawate, and in Ninja Machim Big Titsig Tawate, Betty Loachadores.
And you can imagine now we are very angry because this is a very harmful mutation, which in many cases can be deadly.
That they capture Domba data by the control shoe on you extension was that they that Dawgin injured the Chimba Bana and a cook and injured de la Tenedete and got solar junction again, Netanti Grace.
So while the others we may change one amino acid or may not change at all, on this one we are changing the whole structure of the peptide.
Ku Ninjuga Gama Sim de Nalaya, Atsuga de Casa Emino Chis de Ku Jorduarwa, Never did the Jungoris, that Tamatim by Bana, and you couldn't get Titsig Tawati Copa Kanzala Jorduris.
Any questions?
Try is.
Kana tanda domjo ge mutation la sto de tanda de mishi la de na ya positive ta ya che keben yo ba de yon secret bes.
You said the last one, friendship is dangerous.
Yes.
And is there any is extreme, right? People may die because of this.
Well, it really depends on where.
And
in the sequence.
his question is there a positive?
Yes. So that brings the whole topic of mutations can be good, can be bad, or most mutations are actually neutral. But that is a topic that is beyond this course.
Tanda ko actually yaho cholate yores ko tanda dinner do ninja chimbana kashi yaho kashi duga dinner yores. Tanda ya ta ninja duata do yores. Ninja ma shite ko prani tinda ko khipe me pa chwa me pa shwe me pa tinda ge nethate yores simbana tanda ge chiko ta chwe me pi ge parlo la de ninja ko te tanda nga so thon trishige nana mes.
But one major driver in evolution which creates diversity among organisms is by random mutations creating differences in the genomic sequence of organisms. Some can be good, some can be bad, most are neutral, they may lead to adaptation, and they may lead to evolution of a group.
Daga so chingwe ge nyeri kana leta bai bana penjir rupa ge penjir de ko chongya ge junse so de kare se stambala ya rezi ge na ne nyinjur chong nyinjur la ya ko ta khashi ya wa khashi duwa khashi paro ne de den chi wa shikchen shita me chu chi wa ta de chong la ten de te nga so nyeri kana la ta chingwe ge na la ya na chuk rang jin chong ta chingwe penjir chong de ani ta ngime ge nyinjur se do random mutation wa ngime ge nyinjur chong la ten chong yo res.
But the impact on the sequence is mostly seen in extreme with frame-shift mutations, where the whole structure of the polypeptide changes. Not just one amino acid, but many amino acids. Good. Any other questions?
Yeah. The mRNA pesticide zero. The template the zero. The ine danda dina negur tensa di so the template madem bache coding the gana negur the thing da. The DJ code the DNA strand check the la negur te ba ina jamba de la negur te be capture ves.
Inchila sonda. Chara sonda.
So like normally like mRNA is copied exactly from the template strand.
Yes.
But here the mutation is shown on the coding. So does it
That's just to save space. That's just to save space. If I was to add all 4, we wouldn't have enough space in the sheet. So that just shows the coding, which would be identical to RNA, except we're changing T to U.
So does it mean like when a mutation occurs in a DNA one strand, the other strands also has get the mutation?
Yes, as long as you have the sequence that is being translated, well, transcribed to RNA, you would notice that change. If there is a change in a sequence that is not transcribed or translated, you would not see the change. Right? And that deals with introns and exons in genes. Some parts are translated or expressed, some are not. Okay?
Listen. Tanda kelagite ro tanda ga su de la ya mutation drama tsugo to thama gite ro ne tanda ge coding se ro ga su ga da digite ta cha su du sro ta tanda de gelage ko ta chi sacha te chi ka su ro sacha te ma pu ma sa ge che ta yin ba na re ru ro re ma re te ma chi wa chi sha ja pro dus den de ta ga su go de ka la na DNA ki tila tende ta RNA ti chung korwa re mare RNA ti nang ko ge sangda la tende te nga su gi ta ti tzi ti chung korwa amino acid ti man kore amino acid ti phasji go yorwa re mare this time la ya cham sam la ya ta gela ge sa la ya ane ko ta ninju chung ba ba na yinchi mi chi yong ngong go ya me do re mare ke ti ninju chung ba ne ninju ti nga su ge phasji chi ma chi la ro ninju chung wi ge chashi ti phasji chi ma chi la ra lag do Pajur chebai na tigi shuchen ti tong korwa kete pajur machi bai na ane kungai wa barwa.
One more question? Yeah.
Yache trust insa trust.
Tanda di ngazo peji nyama di nalo la tanda kima la jigdo ge. Peji nyama di la na ni missense di nalo la yan ti di ge gwa di la tene ko ogi dilani tise ta siwayas madle manda ngaba dilani kasana ti ngigir chwa la tene frame sheet khora tar gwa do cheda ta chue di ngigir peje chik dawdo cheda peje nyama dilana chik ma do mde ini ya ko chik la tom khora gwa do chik la missen chada te khande kheba do inas.
He is asking about from the missense.
Yep.
You have T.
Only T.
And in the frame shift also you have which one is over here T.
T.
So we have T the same.
Why do you have a missense and why do we have a frame shift?
Yes.
Because of the combination of the triplet. In this case, we have T, but we also have a G and a C. If you refer to your amino acid chart, that will give you cysteine, right? So this particular combination, this triplet, gives you cysteine. Here, we do not have TGC. We have CTG, which is a very different triplet, giving you a very different amino acid.
Nada de gasuga some centimeters, the some centimeter on bade, and a galaya gasuga TGC, Tilatenete, amino chizimando chungora, water line by Nasulaya, CTG on Gudwa, CTG on Stambala, amino chizimando chungores.
Right? So it's really a matter of the combination of the bases giving you unique amino acids with or without redundancy. You may end up with the same amino acid, you may not.
Also about the first one substitution, the last one is insertion, right? That
yes.
Also makes it, yeah.
Correct. Yeah, in this one, everything, because we are not replacing it, we are inserting it, and we have pushed everything one space. So everything moves one space. And now we change drastically the structure of the polypeptide. Good. Again, at the end of class, go to your muddy points. I'm sure we may have more questions, and Chris will go through them tomorrow morning. So when you go home and there are some unclarities or some confusion, write them on the multi points.
Tanda chisula draw yo pa bana tanda draw yung dos draw yo pa bana ne chela ya chisuna la pepe kapsa ane nga su tondre chisa ge chiwa o te na tele su draw te matro chis ne sanye ge chris gender tripi kapsa tambo te la ya ne chisu topa yo pa ta draw yo pa tuzula lende nangres.
Good. So let's jump, and don't worry about this. This is meant like a pace maker, but that's okay. We're going to start talking about our activity for today.
Ta tanda nga su ge tirang ge chegu ko la ya desi jagi yes.
My friend, can you start passing them around? Thank you. One per student, yes. So, as our friend Chris is passing out the activities, this is known as a POGO group activity.
Da dada de gelagi dada chisula zendala pechutanga ge chegu ge chete teki to kwate te res dada ge tegi chegu de milaka lagala na POGO rukha chegu segres.
So what is the objective of a POGO? And again, as you teach your classes in your careers and your teaching years, you can create activities that are similar to this one.
Da dada jema chesugun zenda thiba da ne chi ya nyam tno li chithya ge dada chi kapsa dada ge chegun dina do chesu damna kores.
These types of activities, which by the way is the same one that I give to my students at Emory, they're meant to make you think critically.
Da tanda de la chegu chithya sajide ku Emory la zenda tripi kapsa ge chegu ta chipa insta dege kari chigre senal hoptu chula ya ani chi samlu ti chi ta karsugre ting sabu tanga ta toksho ge samlu chi draw tanga ge samlu tanga chi gorwa ti ku ke te tegres.
They make you think. They also make you and allow you to solve problems.
Te tambo de samlu tanga chi ge yipate la ya ni kange tinda do pana kange tinda do seji kange ge lens be chi gres.
This is a teamwork assignment, so you'll be working in groups.
Da de kare se stambala nga so rukak je ku chi instance stambala che so ge rukha ngoni de gu yores.
Within your group, each person has a different role, and that entails oral and written collaboration within your group.
Da tanda che so rukha na la ya kare yores na rukha ki me re la ya leka re yores. Tati ane tege kare tong gore se na nga so ge ta kiche she tan dia ni cha nyam de gu yo pa ge ne ta chi yes.
Okay? And also, since you'll be working in groups, you have to manage your time. You have to manage your collaboration.
Da che so rukha ka nyam nyamli chi gu yo sa ne rukha ka na ne chi chu tang zin che tang leka tang zin che la so pa ge tang zin chi chi gu yores.
And you have to synthesize and you have to process all the information that you're sharing with each other and even between groups within the class.
Da tanda tem se che so ge ta su rukha na ne tu du chong ju ge chaten re tem da rukha jenda ge parola ya chaten kari chong yo pate ani du se ise chi gu yores.
So as you are working with your groups, right, think about how all these steps and all these themes are being applied as you progress on your activity.
Da che so rukha ke she gu drupa tal hen tu ani ga la ya kha chi tsha do wa de ne kanda special tang do ti kari kanda s do do ti ko la ya sam ta ro ches.
Because remember, when you're working in a group, not only do you learn from each other, but you also teach each other.
Da che so rukha ke she gu drupi kaps ani kasa rukha ke tam mi jenda sani su la ya ko to sawal ha pa chamares. And the pala yang kukuto sa kutojigo tricky, but susu jama ba pa yang tricky on your race.
Good. So, how do we make a poll work?
That the pogel de korum kanado race.
You're going to be forming groups of 3.
Me sum joy roches.
If we end up with even numbers and you have a group of 2, that's okay. Somebody can have 2 different roles.
Ta chu tanda me sum joy rocho se ta thamatela ya me ni masme pa na ke yo mares ta mechikili minigli kachina degres.
Each person within the group will have a unique role to fulfill within the activity.
Ta rukha me sum joy rukha re na me re la ya chego ti tan de lingen re do ya yes.
The first role is the reader.
Ta rukha tangpote ra likachin tangpote likate karna logni res log res.
The reader will read direction to the group and manage, very important, manage the time that we are spending for the activity.
다 롱이 지어로 롱캔디 가르치고레 시스템 발라야 아니 리카 드베게 람튼 지어로 데 로고레스 테마세 아니 뒤츠에티 당진 지구요레스
We have the recorder.
다 예바티 통고지니스
The recorder will write the answers of the activity and make sure everyone is engaged.
다 통고지니티 가르치고레 시스템 발라야 제군데 탄테르게 드렌티 가자 트라가 렌티 가자 통고지 지nem세 미 차마 코르카난게 르메 차마 주시치 지구요레스
It's very common when we're working in groups. You may have somebody on their phone watching Facebook. So the recorder will tell you, no, get away from Facebook, work on the activity.
다 루카글리카 제 시스템 발라야 산삼이 카시도아 카페야 레네 고탑가 타니 텐다 요레스 다테 메와 셰테 탄다 퉁고 바디게 디그마레스 카프마 살라샤스 모텔라 마타스 레카치쇼 세테 탄스 지구레스
And you laugh, but it does happen. Okay?
제스 케마가고도 와스 테 용거다스
The third role is the traveler.
다 숨바치고로 미 숨바티게 링겐디 카레나 야마 단 드즈바 트래블러 드즈바글리카 티치그레스
The traveler will walk around the room beyond your group to share information and compare your answers with other groups.
단 드즈바데 카치그레스나 장카나나 야마 친로아 아니 로컬 젠달라야 아니 수수게 딜랑카리 임바데 파라야 모토치
Okay? So as a traveler is walking around comparing answers, he's going to report back to your original group to make sure that the answers are comparable and correct.
다 단단 드즈바데 카치그레스나 파도데 타 수수게 로컬 렌티 젠다얌테 모토치 아니 젠다게 텔라야타 소용데 타 렌카리카리 제두티 수수 로컬 데시자 데 답시 제니 제라야 카치그레스나 아니 텔렌티 주 판수 두르데티 아니 텔렌티 드기요 바다 드와지주구요레스
Are there any questions about the specific roles?
다다 그 제구 마보도가 미 디타 콜라야 트라이오스
Okay? Again, if you end up with a group of two, that's okay. One person can do two different roles. And you can just talk about it among yourselves and decide who is doing what role.
타케티 타마텔라야 미니주아 차소나 미치기 니지나드 코레스 미니기 젤리티 지나드 코레스 타티체 아니 첸데체 수양 난쿠난쿠니 두데치데타 미치기 링게 카리드메트 투스타체나드스
Okay? So for this activity, we're going to be dealing with the central dogma, again, going from DNA to RNA to protein. We understand this. We have learned this. We're going to be dealing with different models. I actually drew them by hand. I am not an artist, and this took me quite a bit of time, but I was able to draw models that deal with transcription, going from DNA, making the complement to RNA, and also translation, where you have RNA going into the ribosome, producing your protein chain.
다 단답부터 보텔라야 가수 제고데 레아데 가수 타웨 냥보 간에 팝지다 DNA 니 RNA 라야 팝시체 티지라야 RNA 티지라 팝주체 그럼바데 치고요 레스타 단다데 리무트샤주데 라파트샤이엔스가 리무라야 케바데네 메임바나 데라야 치소 컨센트라지트샤이스 담보데 팝시치 kapsa DNA 기라테네 데테 가수 판첸 RNA 티 팝시치고요로 데라테네 데 판첸 RNA 데 가수 라보타지라 자올라테네 데테 가수 띠나네 아니 지로와다 지라야 파주지용우디 에미노 지라야 파주지용우디레스
You may notice that the activity is actually quite lengthy, and that is the whole point of having different roles, to make it efficient and to make it work well within the group.
다다 제주도 스타바임바네 제주 라발라야 라야기 제구디 림푸 슈치고레스 틴스타발라야 림푸 슈 예자 제출라야 미 숨그나나 미 리리 만다라야 링게 만다 테샤요레스 디스tambalaya 냠르간네 뉴어톰부간네 드랙체트 네 루카클리카 랭보요레 냠르무간네 두과치레스
Okay? You'll be using, again, the coding chart that I made. Just remember, you go from inside to the second tier to the third tier, and then you have your unique amino acid.
다다다가르 조샤주게 데라야 리무치 토가 데페치 땅고레스 다다데야 로타 야시 텐수시 땅바이바나 kimala karichido tigi jila rumba nyola karichido tigi jila rumba sambala karichido tila tene dete ngasula ya amino chirji kare ragadu di tenke gures.
We're going to be using a chart for the different amino acids. So each amino acid has a 3-letter code and also a 1-letter code.
Aha. Tata de la ya ngasu shenda chi amino chirji tang revi gi rumo chi peche tang gores. Amino chirji re la ya ne korang min chamba chi gi ne igi sin chang gi min chi yores. Temse igi chi chi wa gi min chi yores.
So when you have your hidden message and you go from your amino acid to your 3-letter code, also use the 1-letter code to decipher the hidden message that I put in.
Aha, tata de jela ya chi su karichi gure sena ta chi su che gu dribu kabsa amino chirji ne ani tata amino chirji kuran che ko jela ya igi sumchi wa ge ko ge sanda chi to de ga la ngola jir gures te gi jela ya igi chi chi wa ngo te la jir de te ani tanda gela ge che su de na la ya te sha ju ge ani sanda ti kar re ti tre gu yores.
Are there any questions?
Draw ayo sa.
Good. So we're going to end. Basically, the PowerPoint is pretty much over. The talk is over. We're going to spend the rest of right now before lunch. Starting our activity, we'll have lunch. We'll come back and we'll have the rest of the time to work on our activities.
Aha, tata de la ya chi tenya ta tu cha ba yin sro. Ani che su ge zanta de ta ji zoro par ta che gu de ga la ta ro che sa ne nyam ko so chi je la yang shin zong sta ba la ya ne che gu de mut to dre yes.
After today, after we are done with today, I will post the key. So you can compare your activity with the correct answers that I give my students when I teach this class.
Aha, ta tanda tireng ge zanta trenu ge je la ya ne ge la ge ko ge talent yo ro ko ge pa la ya emoril typical se bugizu la yan de chi chi sha yores te ko su ge talent ta yang ta pa ge talent kari imba te ne chi su lo ko to chi ge yes te te kuto chis tamba che su ge talent te ta ne talent ge ju ge la na ju choro o te ni che do se dura yores.
So tonight, after we are home and everything, I'll post the key and tomorrow you'll have access to it.
Aha, tere zenda trenu ge je la tere ngoda ngara ge line du bi ge line te du sha gos ne san che su ge ta yores.
Any questions?
Try is
Does anybody want the key right now?
aha talent duwa ge ta ki sigro ta talent duwa te tanda guji yesa. Talent sama tanda guji ayo sa.
You have to pay me a lot of money for it.
Aha, go pa na kon chamber is special mabote go ke gras.
Good. All right. So yeah, this is over. So just don't forget Muddy points for tonight, for today's class, and Chris can certainly cover them tomorrow. So don't forget to put questions, concerns, points of confusion, okay, anything that you want, put it in the Muddy points, and we'll cover them tomorrow morning.
Aha ya tanda chichu ge ti ma je pa chiro chi chichu chura ta ne chi seu ma chaya ta ya chi tuna chi gu sa tin ra yo pa ba na tra sa chi go wa ti rang te ka chiro chi ne sanje ge la len chong grace.
So for now, let's get into our groups, decide who is in each group of 3, decide who the roles are, put your names on the activity, and start working on the activity.
Aha, ta tampo ta ga su gu chi ge yes tampo te la ya mi sumdi ane ka sa rukha zo ya che en te ge je la ya rukha na ne ane mi su ngen kare demen te che ngen kare demen te tha chen u je la ya te kar sa te ga te le tri sa do wa ra su min te ngen kare de go me te ga tri ane ga su che gu te de yes.
Good. Let's start working.
Okay, ta gu zero announce ane tanda te ge che gu te ge ka sa inchi ge mapila tanji to ba na Google Classroom ma yor da lo te ta ma chi pe na jir sarwa na chung go nyo podo to ku mas ko yo ba na inchi ge mapi te pha jona na yore mi sumde sa da. Tanda Gela Tilen Duba, Len Duba, Sanya Matongi La Sora, or Tirogoda Mategi La Soa. Google Classroom Nashagi, State Jela Tavi Kapsura. Tavi Kapsa, Tilen Duba Tikanza, Tilen Kanza Galaya, and a Tamapo Jabio Resa. The Manana